So we're going to get started. So my name is Marzia Gassimi. I'm a professor at the University of Toronto in computer science and medicine and a faculty member at the Vector Institute. And I'm very happy to introduce to you today Dr. Nigam Shah. So Nigam is a faculty member at Stanford. But when I first met him, I was a student at AMIA. My advisor introduced me to him. And he immediately gave me really good, really relevant advice. And actually just now in the hallway, he also gave me very good, very relevant advice. He said, I looked at your course website. It looks like a first-year faculty website because you had 3 assigned readings per week. And I thought, isn't that what we're all supposed to do? He said, no. So he's both been a very good personal friend, and he has a lot of wisdom. And his, when I introduce people, I like to tell a funny story about them to humanize them. And the funny story he has given me permission to tell about him, there are many, but this is the one we're telling, is when he was applying to programs, he applied to several places, right, for doing his MD. And he applied to Penn State and a bunch of other places, and he ended up going to Penn State. But he had also applied to Baylor. And when he got in there, he chose not to go there because he thought, oh, it's Baylor College. I don't want to go to a college. I want to go to a university. And so it's important to know what those names mean in different contexts. So without further, one more thing that I was told to say about these in general. So you guys have name tags. If it's pink, it's because you're on the machine learning side. If it's blue, it's because you're on the clinical side. And the goal of these events is to have mixture between people who do machine learning and who do health. So talk to people with a different colored badge from the one that you have. And our next health AI rounds for Vector will be on May 6th. You can register for that on the Vector website. So now, without further ado, let's give a round of applause for Nigam.
Thank you. All right, am I audible without the microphone in the back? Good? All right, so that way I can wander around a bit and so on. So thank you for having me here. Deep learning has been a distinctly Canadian enterprise, and it's a great pleasure to be presenting at the Vector Institute. And I did not know it's right next to the Toronto General Hospital and like a block from the Sick Kids Hospital and so on. All right, so I'm going to tell you 2 broad themes of stories today. Which is in the talk abstract, which I'm absolutely sure everybody read. But there's only 2 stories, and we don't have to get through all of it. I would like to take questions along the way rather than save them towards the end. So that way we get about 45 minutes of conversation as opposed to me talking for 35 minutes and then everybody having to remember the question from slide 3, which is probably not going to work. So feel free to interrupt me at any time. And let's get started. So let's meet Vera. She's a 70-year-old Asian woman with a history of hypertension and asthma. She's on metformin, but now has uncontrolled diabetes. So of the blue tags in the room, how many of you identify with such a common occurrence? Yeah, it's a fairly routine situation. I saw a hand in the back. And just what's the rough sense of the ratio of the blue tags? How many people are clinical or self-identify as clinical? Blue tags, just a quick raise of hands. All right, and okay, so about 40%, thereabouts. And so we get cases like this all the time. In fact, the case that inspired most of what I'm going to tell you about was a 13-year-old female patient with systemic lupus erythematosus. And we were tasked with having to make a decision about anticoagulation for that case. Today I'm using a sort of a sort of less dramatic example. So how might machine learning help Vera? So if I break it down into the essence of the matter, in this 15-minute or 11-minute encounter in the US, a clinician is expected to assess her risk of a stroke, risk of a heart attack, a risk of renal failure, ophthalmic adverse events, and figure out how to manage her uncontrolled diabetes. and do that while looking at the patient and being extremely empathetic. It's kind of a tough job. So in that process, a lot of these risk assessments have to be done. And if the risk of something is above a certain threshold, a different action would be triggered. So if the risk of heart attack is above a certain threshold, maybe we should start that person on a statin if the lipids are out of whack. If the risk of renal failure is pretty high, well, we shouldn't be on metformin and should be treating with something else. The problem is that in any given situation, 10, 15, 20 of these apply, and each one of them, the guidelines or the guidance in textbooks, is produced or manufactured in a very pristine situation where nothing else is going on. But in real life, you have to compile all of this in your head, make a decision, and move on. And then hopefully, in an urban setting, you probably record that in an electronic record system, but we never go back to look at that. So when I started thinking about this situation, I thought, well, if I look closely, this risk estimation is either a classification problem or a prediction problem, or diagnosis, prognosis, if we speak the medical language. And then the action is informed by the estimation of the treatment effect. In most cases, the average treatment effect or the population treatment effect, and depending on the situation, maybe there will be words like individual treatment effect, personalized treatment effect, or a heterogeneous treatment effect, or CATE, conditional average treatment effect. And different disciplines will use different words. But for now, at least for this conversation, let's just pretend it's a population level. So if we figured out how to do those two things, maybe we can help Vera. I know she's not hypoxic right now. It's just the projector that's showing the lips blue. As a doctor, you see blue lips. It's like something is wrong. No, I think it's okay. All right, so the classification, prediction, and then treatment. So classify, predict, treat. Three things I can do with algorithms. And I don't use the word AI anymore or machine learning anymore for the most part, because at least in a clinical audience, AI was invented in the 60s. It has a CPT code, and it's for artificial insemination. So it doesn't go too well, and mostly practiced by veterinarians these days, at least in the heartland of America. Now we can use algorithms to solve classification, prediction, and treatment estimation problems to advance the science, the practice, or the delivery of healthcare. And this is a crucial difference, particularly for the people with the pink tags. And I'll have examples. We're not going to talk about the science examples today, but the goal there is you use algorithms to advance the understanding of diabetes. Like, how does diabetes happen? You're typically dealing with sample sizes in the 500 or 1,000. You have genomics, metabolomics, proteomics, and the goal is to find that next elusive drug target that Eli Lilly will then go fund you to market and do a trial for. I don't know whether Eli makes any drugs. I just read their name downstairs, so I use that. On the practice side, you want to be using algorithms to do things that improve clinical care today. The first column is about advancing understanding of the science that allows us to come up with new therapies that improve care sometime in the future, usually 5 or 15 years in the future. And then the last column is about the business of healthcare. I'm a doctor, I have a panel of 5,000 patients. How can I use algorithms to make sure that the 2 people I need to call today get called and I discuss the thing I need to discuss with them? It's customer relationship management and not really science. I mean, there's behavioral science that goes into it, but not the molecular biology science that goes into it. So I spend my time mostly in the middle column, trying to use algorithms to advance the practice. And it's sort of a hedge, because if you're in the first column, the chance that you advance the science and end up advancing the practice is maybe 1 in 10, maybe 1 in 50. And if you advance the practice, the chance that you advance the business or delivery is again 1 in 10 or 1 in 50. So the biggest mistake that could be made is someone spending 5 years trying to advance the science using AI and then discovering that it doesn't advance the practice and has no relationship whatsoever with the business of healthcare. And that happens over and over and over again. So I just wanted to show this grid that as you bring these two communities together, be clear what you want to be doing. I mean, all of these 9 cells in this grid are perfectly valid scientific enterprises. Just have to set your expectations right. And the funny way of looking at it, the first column you publish in the one-word journal called Nature, Science, and Cell. The middle one you publish in JAMA, New England Journal, and Annals of Medicine. And the third column, you kind of don't publish, you start out a company. Or maybe you publish in Health Affairs. So that's sort of the cartoon way of thinking about it. Okay. All right, so that's the only sort of didactic big picture slide I have. Now let's get down to specifics. So here, I said I'm going to talk about the middle column. We'll be talking about one example from here and one example from here. Probably not going to talk about a classification, but the principles remain the same. So my research team, we were about 20 people. We deal with the electronic health record repository of Stanford Health System, about 4 million patients over 8 years, 10 years roughly. And then we have access to claims data from Truven and Optum, stuff you can buy in the US, and that's about another 200 million lives over 8 years. And then using that, we do 3 buckets of projects. The first 2, Informatics Consult Service and the Program for AI in Healthcare, are the ones mentioned in the abstract. Those are the ones we'll talk about. And we do lots of funky stuff about producing insights from non-conventional data sets that we will not discuss today. I'll just leave you with one tidbit. The last one we're doing, most recent one we're doing, is looking at 900 hours of psychotherapy sessions and the audio recording and the corresponding PHQ-9 score, the depression score, and trying to learn what are characteristics of a good psychotherapy session. So that's pretty wacky stuff. And more about this at this particular URL. All right, so let's start out with the Informatics Consult Service. And I'll give you a little bit of a background on how I arrived at it and then what the service does and so on. So remember Vera? She came in with diabetes. So let's just work with diabetes as an example for today. So the first thing I want to know before deciding on a treatment for her is, how is diabetes treated today? So this picture is from a paper led by George Hripsack, part of this observational health data science and informatics community, where a few people from Canada are involved. It's about 90 to 100 sites, loose collaboration. It's not a legal entity. And we studied 3 diseases: diabetes, hypertension, and depression. What we're looking at is diabetes. The first circle is telling you the first drug that people get for managing their diabetes. The second circle is the second drug, third, fourth, so on. The color tells you which drug, which is kind of hard because most of it looks blue when you take the red pin out in RGB. There's more colors here. So most of the time, people start with metformin, which is as it should be. And when they don't start with metformin, there's a clear reason: they have poor renal function. So life works as advertised, actually. People, by and large, are following the guideline. And this is based on about 250 million patients worth of data. But then, like Vera, they come in, their A1C is not controlled, they're already on metformin. I could go here, here, or here. You can only go to the first one in Britain because they're the only one that have glycoside. We don't have it in the US or North America. Which of these 3 are better? The guideline essentially says, pick a drug. That's what it says. Pick a second-line drug. There's 34 randomized trials that attempted to answer the question, which second-line drug, with no conclusive answer. None of them have had anywhere close to 250 million patients. So we did that study as a follow-up, and it took us a couple of years to get to that. That one I think was 2016, and 2 years later we found the answer that compared to sulfonylureas, which are the most common ones, DPP-4 inhibitors will get you a faster time to getting your A1C under control, and not shown on this plot, lower rate of adverse events, slight smidge lower. Okay, so that's an answer. I can use that to make a better decision for VETA. So this paper came out in JAMA Network Open. In a rare fluke, another group was studying this exact same problem, and completely independent of us, they came up with the exact same answer. So that was awesome. When that happens, you get to write an opinion about it. So we wrote about a second opinion from observational data on second-line diabetes drugs. So in this case, evidence was manufactured and delivered on the classic medical profession life cycle of 2 to 3 years. It will get into a guideline. Maybe the practicing doctor will look at it. Maybe the CMIO of your hospital will implement that in your medication ordering system, or maybe not. A press release happened and all that good stuff, but not sure whether it's going to really change practice. So this is one of many other studies like this that we've done, looking at androgen deprivation and the risk of Alzheimer's, and silastazol and major adverse cardiovascular events, and uveitis in kids, and so on. But 15 or so papers between 2011 and the last one was 2018, but let's say give or take 2015, 16, got me tenure, so that was awesome. But the case that stuck with me was one where a pediatric rheumatologist was tasked with having to treat this 13-year-old with systemic lupus. She had a cohort of 98 patients, which she had access to for research. Based on that, she concluded that, or did some very simple, keyword-based matching, and found that if the kids have high proteinuria, they have a high risk of coagulative problems, and so she chose to use anticoagulation. That patient was fine. She made the leap from a risk estimation that high proteinuria is high risk, so risk is above a mental threshold. Therefore, take action. In this case, the action was anticoagulation. The action was never tested, or its efficacy never estimated from the data. That got her a paper in the New England Journal, and also got her in trouble with her CEO, legal, dean, compliance, privacy, like IRB, everybody, because she did something shocking and irresponsible, which is to use data collected for research to improve patient care. And before the press embargo lifted, within a span of 24 hours, all the senior folks sort of got together and thought about this and said, wait a minute, this feels wrong. This is why patients come to Stanford for. Do you not want the best evidence from prior patients informing the care? And so they all signed off on this effort post hoc, except the IRB, which gave her a slap on the wrist, saying you should not have done that because you said you would only do research, but lo and behold, you took good care of a patient. Like, how's that right? So I was on the faculty right around this hoopla was happening, and I was doing all of these studies, and that led to, we shouldn't be doing, a doctor should not be expected to do this in their garage. We don't do a kidney biopsy at home. We don't expect a primary care doc to sort of have an MRI machine in their backyard and say, hey, come on over at 8 p.m., I'll do the MRI for you. It's a systematic service offered by the hospital. So that's what led to this informatics consult service idea, where given a complicated clinical situation, we would want to biopsy our database and produce a report of what happened to patients like mine or similar patients. So these two URLs will get you more information about that. The first one is a 1-minute video. And so let's start with what the service gets you. So here's the real report. This question came asking, do patients who have an mildly elevated, incidental, mildly elevated first kappa or lambda free light chain test result go on to develop malignancy? This is copy-pasted as is, very colloquial speak by the clinician. We then take this question and expand it into a question that can be asked. This is how doctors speak with each other. You can't speak like this with a database. So we do the translation from this question to something that can be asked and answered off of a database. We do the analysis, and then we write the interpretation. Just like you order an MRI, the radiologist reads it and gives you an interpretation, or impression as it's called. We give you an interpretation, the bottom line. And before giving the interpretation, we actually speak with the clinician saying, what is the real decision you're trying to make? In this case, the real decision was when to have the patient back. It's a low-risk decision. Given that mild elevation of this particular test is associated with development of hematological malignancy in 6 to 12 months, it is okay to say, see this patient again in 6 months as opposed to 2 years. You don't have to have precise statistics for this particular decision at hand. So the key information here is this iteration with the clinician, that what is it that they're actually trying to do, and then customizing your analysis in order to facilitate that decision as opposed to having some absolute pristine version of the truth that you can go publish somewhere. So there's two slightly different things. And then if they're interested, they can go in and look at the cohort demographics, look at table, gender breakdown, age and so on, look at Kaplan-Meier plots and the statistics and the p-values and all of that stuff. So that's the idea. We've been offering this service since late 2017. We've done 135 cases so far. And the process involves 4 humans: the requesting physician, an informatics trained physician, an EHR data specialist, and a data scientist. So the first thing, the doctor makes a request. The informatics physician, his name is Saurabh Gombar, I'll show his picture in a minute. They make sure, are we asking the right question, which is going from that one line to that full paragraph that I showed you. Because many times the question might be phrased as an inferential or a predictive question, but in a 5-10 minute conversation, you can figure out that a descriptive analysis can still produce useful information for their decision making. And descriptive analyses are way simpler to do and a lot more defensible methodologically than inferential analyses. Once the question is settled upon, we do the definition of the cohort, the exposures, and the outcome. And we'll dive into that in the next slide. Typically, that step, getting your data cut with diabetes defined as you would want, is anywhere from a 2-day to a 6-month adventure, depending on the institution you're at. We compress that down to a few minutes. Then you build a cohort. That's a few milliseconds. Once you have that, we spend the majority of our time on the statistical analysis, both clock time and human time. And then after that, a report gets written. Most of it is automated, but then all of these 3 individuals look at it to make sure that it is something that they're comfortable sharing back. These 2 people write the interpretation, which is then shared with the physician, who can then take the appropriate action in response to the information we received or we provided them. So let's drill down into this particular piece, creating the definitions and building the cohort. So when you say, and the whole process takes about 24 to 72 hours. It used to take longer, and as we iterated and we started making things better, it's a service. It's not a software tool. And it's like running McDonald's. You have to figure out, how many minutes am I going to take for this particular task, and so on. So when most people think data, if you're a clinician, some version of this thing comes to mind. Depending on the particular IT system you have, the more expensive it is, the uglier it looks, by and large. Usually a whole bunch of structured fields and so on. And then, of course, the clinical documents, which are haiku of acronyms. I can see the clinicians are all identifying with it, or the people on the data mining side who've suffered through it. I was like, seriously? I thought natural language processing would work. But my point is very simple. Natural language processing works on language that is natural. Enough said. So when you think of data, I want you to imagine a timeline. I don't want you to imagine a picture or a pile of notes or a data table. Imagine a timeline, because healthcare happens over time. So the gray arrow is the timeline. The gray circles are when things actually happen to the patient. And then depending on who you are, some version of what happened will be recorded. So disease codes, medications, procedures, imaging data, whatnot. If you're in the ICU, some streaming data will be collected. So think of a patient timeline object. And we'll have that object for multiple humans. The challenge is to go from, now this object or objects are stored over 100 different IT systems. There is no one place where you have all of it. The challenge is to go from that to a tensor that you can deal with. And then after you have the tensor, you still have this phenotyping problem to say, I will believe somebody is diabetic if they have the ICD-9 code 250.02, they have metformin and an A1C value that's abnormal. And that's the phenotyping, or electronic phenotyping. For the informatics community, phenotyping is the declaration of the necessary and sufficient conditions to believe a patient has that condition, exposure, or outcome. The computer scientists have a slightly different version of phenotyping, and learning representations is also called phenotyping. That creates a decent amount of confusion amongst the blue and the pink badges. So just an alert on that. And then you have to make decisions about how will we handle time, what features are we going to use, and how are we going to state the phenotype definition, which is the necessary and sufficient conditions for you to believe the patient has diabetes, or they had a stroke, or whatever else is of interest. So we solved all of these problems by one piece of software. And we made a whole bunch of decisions, such as inventing a new domain-specific query language, inventing a new search engine, and we did not develop a user interface. The user interface is a command line. For a very good reason, because if you build a user interface, then there's this slippery slope of, oh, let's make it friendly enough that a doctor can use him or herself, and I'm not a UI person, and I'll just leave it there. So this is the interface we use. We never show it to a doctor. This is the command prompt, basically. You write in a syntax that looks freakishly like Lisp. And in this screenshot, it's an old one. We're querying 2.3 million patients. You can query codes, notes, lab results, vitals, and so on. And then once you have the cohort, remember I made a big deal about the patient timeline object. So we're looking at multiple patient timeline objects. One of them is highlighted, which you can't see. And the colored portions are what fragments of your query matched what portion of the timeline. And then you can see everything else about one case, which is selected here. And the point of looking at the data in this way is that almost every time we look at this, we go back and ask the clinician, your query or your description is pulling out these kinds of patients. Is that really what you wanted? It's like, oh yeah, I forgot about this, I forgot about that. We're able to do this iteration in under 15 minutes because this is a live system. It's query as opposed to a request you fill out in a drop-down menu and a form, and then you get a message 2 days later. That rapid turnaround is absolutely essential, and it avoids so much problems later on. And so there's a video of this search engine at this URL where you can see the syntax and how it's performing and it working live on a 200 million patient data set and so on. Okay, so in the pilot phase, we've done 135. I made this plot a little while back. 55% of the questions we got were descriptive questions. That was actually eye-opening for me. I thought most of the questions would be inferential questions, like which drug should I use? That's not what the majority of the questions were. The majority of the questions were, how often does something happen? Like they already know I'm going to do X, but if I do X, how often is ABC going to happen? And then because we do an in-person debrief, once we give them the report, we ask them questions about, did the interpretation section make any sense, and did you like the cohort demographics and so on? And one of the questions we ask is, what are the top 3 words that come to mind when you receive the consult? And this is a tag cloud of the top 3 words that 135 clinicians said across multiple questions. Actually, no, 70, 80 clinicians across 135 consults. So on this plot, on the x-axis, we have the number of physicians requesting a consultation. On the y-axis is the number of consultations, and the circle tells you the specialty and the volume from that specialty. So internal medicine, about 23-ish unique doctors have requested about 35 unique questions. And we've gotten questions from practically every specialty. We make a lot of efforts to make sure that we're not wrong. I don't care as much about being right, but I don't want to mislead people. So we use these things called negative controls for empirical calibration. For the clinicians in the room, just think of it as associations that are blatantly absurd, as in aspirin associating with left foot fracture but not with right foot fracture. And then test 100 of those. And the fraction of that for which your analysis says thumbs up, true association, well, that's the extent to which you have a problem. And then there are these things called e-values, which give you an analytical calculation to say, if there was confounding, how strong would it have to be to produce the effect size that you're seeing? So again, this is to give some guidance on, if there's very low degree of confounding that it can produce that observed effect, then maybe you don't want to trust the results. We ask the question using multiple data sets, and we schedule an in-person debrief so that the numbers as seen get interpreted in the safest way possible. If you want to prank your medical MDs, medical friends, there's a nice paper by Zach Coheney on medicine's inconvenient relationship with math. Beautiful paper, guaranteed to piss off the clinicians, but it is very eye-opening as well. I think a good 95 or 98% of medical professionals couldn't get the positive predictive value of a test correctly interpreted. So that's why we do the in-person debrief, because in a high-paced 15-minute decision-making situation, you don't have the mental bandwidth to sit and reflect and say, what does this number mean? It's much better if someone does that interpretation. So here's the team that runs the show. Saurabh Gombar is the informatics clinician. Alison Callahan is the EHR data specialist. Vlad built the search engine. And Ken Jung is the data scientist. And then we have partners on the hospital side, outlining the business case, making sure the MDs have a clear way of ordering the consult and receiving the report. Topher Sharp is our CMIO. And David Entwistle, our CEO, is also quite supportive of this project, because it is about preeminence. It's about being first, which is what Stanford likes. We hate being second. So we're the first academic hospital to have such a service where we can produce a report on what happened to patients like mine. Okay? All right, so I'll switch gears a little bit, and we'll go into this program for AI in healthcare. And these 2 URLs will get you more information about that. Yes, questions?
So for those questions, are you just looking at electronic medical reports, health report, or are you looking at different research articles, clinical trials?
Only medical records and claims. We're not doing the equivalent of a library case lookup. That is something that actually Alison Callahan trained as a medical librarian in Ottawa. So she's very interested in bringing that piece in, which historically all medical centers had. You had a medical librarian who went on rounds and would look up the cases and produce a report. But we don't have it right now. It's a great question. Yeah.
Obviously, this is a very useful tool, and you have a large database to learn from, right, to produce your answers. I'm wondering, what are your plans for the extension of it? Further development of this tool, especially when we take a look into genomic data, which is coming. We are aware that all these millions of people don't have yet. but they are coming, right? So genomic data definitely give you additional insight into conditions and potential interactions, right?
Mm-hmm.
Analyze genomic gene-disease interactions, access public databases that already collect all these results of different research and so on and so forth. So are you planning to extend your methods to actually analyze genetic data and interactions that will be more personalized?
So not quite genetics data, because when I talk to these guys, they basically say what I really want is the SNP, or single nucleotide polymorphism, for patient satisfaction. I mean, it's a joke. That doesn't exist. And so far, the use of genomics for better clinical decision making is hypothetical. It is still on the science side. Measuring 54 loci to estimate diabetes risk is just not worth the cost. I can measure height and weight and ask whether your father had diabetes, and that's a much better predictor than the genetic information. Genetic information does have utility in tailoring oncology therapy, for example, for dosing of about 30 or 40 drugs. But if you look from a health system perspective, the number of people that need to be acted upon to realize benefit for one human, the math doesn't add up from a system level. So while it's great science right now, the translation to clinical practice and health delivery is still lacking.
Does it mean that you think that practice side is more at, let's say, evidence medicine level, and science side is more at precision medicine side? Because we can't speak about precision as it is if we don't have that input.
Yeah, so
it's more evidence medicine, not precise.
Correct. By and large, current medical practice could be called evidence-based practice or opinion-based practice, if you want to be very truthful about it. The whole idea of precision medicine is a little bit of oxymoronic. It is a rescue job on something called personalized medicine, which didn't work. And if you think about it for a second, the more features you try to match people on, the fewer matching people you get. And so every time you add more dimensions on which to match people, you will be basing your decision on fewer humans, and your variance will increase. And until we make advances in the mechanistic understanding of disease and have true causal models of the underlying biology, personalized medicine or precision medicine by the application of supervised learning is unlikely to work, because you'll always run into this trade-off of having too few humans or too few samples from which you need to make inference. And it's just like basic math problems you run into. It may be solvable, but I'm not too optimistic. What I am optimistic about is the development of causal interpretable models of the underlying biology, which then allow you to do true personalization. And that's starting to happen in the oncology space, that given the specific tumor profile, you can predict which drug will or will not be effective on that tumor, and you can use that to guide therapy. So it'll happen, but I would say majority is still in the science side.
I was wondering sort of what you have seen as the willingness of physicians to accept results that were counterintuitive to their own intuition of based on practice.
It's a great question. I would say half of the challenge is the culture change to allow data to have a vote in the process. And medicine has been trying to do this. So this informatics consult service, sounds brilliant and fancy and new and all of that. The first time somebody did this in the United States was in 1972. at the New Haven Yale VA. 1972. The downsides they had were their library of patients was 678 patients, so not too big. And the disease that they were trying to give recommendations for was lung cancer that didn't have many good treatment options in the first place. But they did the exact same thing that we did. They had a knowledge graph. They had interactive query language, iterative personalization of the similar patients, full thing, end to end. The next time somebody did this was in 1978 timeframe at Duke. There the difference was they did it just one service line, cardiology. The third attempt was at Stanford, just rheumatology. The main difference we have, and all of these three projects kind of got stalled because of data availability, because electronic medical record penetration was abysmal back then. So we just happened to be at the right time, so pure dumb luck at some level, that there is enough data, there's enough compute, that if we do those exact same things people had done in the 70s, now it works. Yeah.
So how do you see this scaling up? Do you think every hospital would have an informatics consult team, or could it be a centralized consult team that hospitals would contact?
So that's a great question. That's what Tip and I are currently struggling with. So you could view this as a specialty service line and train people, and then every hospital would have its own informatics clinic, so to speak. That's one model. You could imagine a central facility like a reference lab that's doing testing for everybody. So it could be done at a national level, for example, or a provincial level, for example. It could be that this doesn't belong in a company and it should be like GE making MRI scanners and you just spin it out as a company. We don't know what's the right answer yet. Yes?
So I think what you've built is incredible, but I think your point about worrying about the predicting wrong is clinically would be the way that I would worry about it as well. And there's examples from observational data that have had hundreds of studies examining the question, and I think the best example in my mind is hormone replacement therapy, where there was hundreds and hundreds of studies, and some of those studies, like the Nurses Health study, had hundreds of thousands of participants, and prediction after prediction after prediction got it wrong, saying that it was a beneficial treatment for women, to the point that it was the second highest prescribed drug in North America at this point. And then finally that randomized evaluation happened, the randomized trial, which showed that the causality was in the opposite direction, that it was harmful. And I worry that with this kind of mining, and given the subtlety of effects and the variation in the kind of patients we're applying to, that there's a lot of potential to get it wrong. And I wonder what mechanisms we would put into place to protect against that. And one idea I would maybe suggest is that there are examples where there is randomized trial evidence out there. And I wonder if your prediction models, if you ran it in your database on a purely observational data set, agnostic of what the trial showed, would get it right. And how many times would it get it right?
Yep. So that almost always comes up. I promise you this was not a plant. This is what we do to not be wrong. The challenge with the reproducing the RCT, so to speak, is that so all RCTs complain that they have trouble recruiting, and only 3 to 4% of the population is eligible. So if I emulate an RCT, I'm only recapitulating evidence that is applicable to those 3 to 4% of the population. So I've basically accomplished nothing other than showing that my observational study is quote unquote good. But we do that just to satisfy the obvious concern that how often are the things that are right, do I pick them up? So in 2013, we did a study looking at the drug recalls, for example. And 7 out of 9 drugs recalled, we could have picked up 2 years ahead of schedule. So that's the one answer I have. We haven't done an exhaustive RCT recapitulation for the reasons that you usually just don't have enough patients, which is the whole point of having such a service. So that's why we focus on quantifying the chance of being wrong rather than trying to quantify how often that am I correct. So it's not a great answer to your question, but that's the best I have right now.
One question, please, just to clarify. In your software, actually, the lady at the beginning, in your example, she has more than one disease. And now when in your software actually risk, she can have more than one risk. And I'm wondering, does your algorithm take into account that there are more than one disease and more than one risk?
So the software is just building the cohort. It does not do any statistics. It's a search engine. The analysis can be set up as a competing risk analysis if the question and the situation demands that. So that is handled at analysis time as opposed to cohort generation time.
But regarding complexity of the problem, I think an interaction between diseases and risk.
Yeah, so that depends on the requesting clinician. So for example, a recent one, kids with cancer on chemotherapy present to the emergency room with fever and they're non-neutropenic. Should I give antibiotics or not give antibiotics? Some clinicians are okay with just building the cohort in this way. Others would say, no, I need the simplified SOFA score, which is based on heart respiratory rate and blood pressure, in order to make sure that I'm looking at equally sick patients. And so depending on the situation, we can pull out those things and the vital signs and use them in the analysis. But that's handled on a case-by-case basis. The software doesn't require you to do anything. So if somebody asks a dumb question with just two variables, the software does not prevent that.
What's also interesting, drug interactions. Are you looking in I'm aware that Stanford has a good database, research database of drug interactions, and also we have in Alberta, drug bank and all this stuff. So that's very important part of defining the therapy. So are you doing analysis of drug interactions and trying to recommend therapy that doesn't?
I used to. I think a lot of people in the U.S. know me as a pharmacovigilance researcher. I stopped doing that because I couldn't find a way to change physician behavior based on what I found.
Okay.
So
Fair enough.
so that's the truth.
That seems important aspect. Or precision, whatever you call it, but it's coming.
Yeah.
And your first sentence is saying if this.
Yeah, I'm not saying
this is your therapy. Right?
It's an important problem. I spent 5 years on it. We even released a database of, top most common 5 drug combinations. Can't figure out how to close the action loop. Yes, in the back.
Yeah, I had a question about this is what we just talked about. So you mentioned that over the course of a year and a half, you did 300 consultations?
135.
135. Is that a lot? Is that below what you expect for the doctors uniformly using your service in a whole hospital? Like, are some doctors reluctant to use it for some people?
So this is all word of mouth. We're not advertising much. And we're actually asking the physician to overcome a pretty high activation barrier because they have to talk to us for 15 minutes. And that's partly by design because we don't want people just asking curiosity questions and just waste our time. We want people who are committed to knowing the answer. And if they're willing to pick up the phone and spend 15, 20 minutes, for a busy clinician, I think that's enough proof for me to say, yeah, they genuinely care about the answer, and I'll spend the day helping them out for that. Yeah.
This is kind of related to how not to be wrong. Can you elaborate on your protection against malpractice or other liability? Because once you produce a report, that can influence a decision made by a physician. How is this service protected against legal issues?
Yeah, so right now it's under IRB, so it's research. The legal precedent always in this case is that the information you're providing is advisory to the clinician. So the liability in the legal sense rests with the ordering clinician. Because that's the precedent for anything. You order an MRI, you order an angiogram, it rests with the clinician. Yeah.
Like others have said, tremendous work that you're doing. This slide that you've left us with, it begs some questions and curiosity. The e-value piece is quite interesting. It's like the one sort of quantitative part of what otherwise is a qualitative approach to try to explore kind of false negatives, let's say. So that idea of, so what's your experience with that aspect of the impact of confounding in the context of these observational studies? And certain aspects of your data, it sounds like big data, where variances tend to be quite small. So the biases could be more the confounding, the selection, the measurement, that kind of thing. But e-values, or that idea of sensitivity analyses to explore the potential effect of confounding, the standard observations in observational studies has been, it's really size dependent. So like with small clinical series might not be that informative, heterogeneity, that there might not be that much variation in a specialized clinic. And then basically it's therefore N, sample size, and strength of association. So can you comment on what you've been seeing about that E statistic in that slide?
I don't have a large amount of consoles on which to base that, because only a third or less needed that. So I wouldn't make any generalized statement yet on their utility or lack thereof. I would say ask me again in a year. Okay, so we're at time.
Yeah, maybe a couple more minutes. Do you want to close here, or?
I could close here, or I could share 3 slides on the actual predictive modeling in healthcare.
That sounds great.
Okay. So this is the cast of characters involved in the program for AI in healthcare. You can see, business and CEO are both involved, and then a lot of hospital IT came in, a lot more clinicians came in that. So here, I'll zip through 4 slides, because there's one bullet point I really want to get across, and I'll start with one example. So palliative care in the US and in Canada, which I confirmed at McGill University General Hospital, is too little, it's too late. So not enough people get it, and those who get it less than ideal time before their death. Median in the US is about 2 days. It's one of the few interventions that have been shown in randomized control trials that if patients receive palliative care, they have a better quality of life, they live longer, and they cost less. So it's an intervention we know works, but we don't give it to enough people, and we don't give it to them soon enough. So I'm going to skip over all of the shock and awe numbers. And so in our case, what we said is, how can I increase the number of people who receive palliative care at the right time? That's the question we started to solve. And so the key construct I want to plant in your thinking is this notion of a prediction-action pair. So ideally, I would want a classification that says this case will benefit from palliative care, this case will not. I don't have that label training data. So we cheat. We use a surrogate outcome. We pretend saying anybody who died could have benefited from palliative care. But then classifying death is not useful, because then they're already dead. So now I have to solve a prediction problem. And we built a predictor to say who is likely to die in the next 3 to 12 months. So why 3 to 12? Because from the action perspective, if it's more than 12 months, people are not ready to have that advanced care planning discussion. And if it's less than 3 months, believe it or not, the logistics of the healthcare system can't move fast enough to give them that the benefit of that conversation and planning. So that's why 3 to 12. So if you have a prediction date, you have some observation window, you got to think carefully about what you're going to include in that observation window. If you have lots of papers around the size, the scope of features in that observation window, how do you do feature engineering and all of that stuff. And then the action window, I already told you, and the lead time in this case, minimum is 90 days. And then we built 3 models. Well, 4 models, because there are 2 models here. The most press is for this one, the deep neural net. Has not been deployed. The things that are deployed are these and the last one, the recurrent neural net, which produces time to event. And so then the studies which we're doing, Well, first we just did some technical validation that you train on historical data, say up to 2015, validate on 2016 and 2017, and okay, the algorithms work, thumbs up. Then we tested this assumption that I'm building the class predictor for a surrogate outcome. So we run this every night. On the left is our School of Medicine where we do all the modeling. Every night, about 80 or 100 patients get admitted. I get their nightly feed by about midnight or 1 a.m. We run the models. We write the results back into the data warehouse by 6 or 7 a.m. And then a report goes out to the head of palliative care in the morning with links to their medical charts. The reason to do it this way is that we first did that silent deployment, and we're tracking how often is it the case that a medical record flagged by this surrogate outcome of mortality is deemed to be good enough to say, yes, we'll benefit from palliative care. And turns out that the agreement is pretty strong. So a nurse went through about 150 records and a physician went through 150 records. Algorithm is predicting, they look at it, say, yes, looks good for an advanced care planning discussion. Okay. The second thing now we're doing is I know that their capacity is about 6 to 8 more conversations a day. So I don't want to optimize PPV blindly. I want to make sure that PPV at 6, as in my top 6 results, are correct. Because they can only call 6 people or 8 people or what have you. And as their capacity increases, I can keep dialing down or dialing up that threshold. And instead of just optimizing for overall AUPRC or AUROC. So now I'm going to zoom out really quick. We have seen some version of this timeline before. And what I want to leave you with is this thinking about as you apply machine learning in health care, we're not talking medicine, we're not talking science, we're talking health care. By and large, the applications tend to be intra-encounter, which is at a given encounter, you look at a picture and say there's pneumonia or cardiomegaly or what have you. Or within an ICU admission, you look at multiple data streams, maybe even text, and say person will crash in 24 hours and you'll need a rapid response team or what have you. I think people do this because it's about automating an existing task and it's easier to imagine what might you do with it. But what's really needed to move health care is to look at all the data, including genomics, socioeconomic, social determinants of health, all the prior data, to make a forecast, a true prediction, with enough lead time so that you can do something different. If the prediction does not offer you the opportunity to take an alternative action, it is a useless prediction. I can predict all of you are going to die someday. 100% PPV. Completely useless. So think about connecting predictions to actions. Each of these rows are the different models we've built, and each of the columns is a stakeholder who will go ahead and do something based on my prediction. And don't build models without talking to them, because the properties and characteristics of the action have a much stronger influence on the final utility of your model, whether it will be perceived as fair, whether it's perceived as ethical, than anything you do on the model side. So I'll stop there. I had a whole bunch of stuff on ethics and utility, which we're not going to go into. If I can take 30 more seconds, I want to show one slide for the pink badges. Okay. So as you build models, and I'll go very fast, you can do representation learning or you can do hand feature engineering. Right? Everybody with me? The pink badges can, the blue badges can check out, no problem. It's not going to matter. And then when you test models, here are inpatient mortality, long length of stay, 30-day readmission. I'm using counts as features, or normalized counts, and a learned representation. This is the typical result you see in a lot of papers, right? That about a 10-point improvement on a bunch of things. We did a very simple experiment where instead of the final model being a penalized regression or something, let it be a gradient boosted model that can model nonlinearities. Oops. So I don't have to do iceberg melting amounts of compute. No GPU needed for this. As you publish in the machine learning community, use realistic baselines. This is a fake baseline. All right, so with that, I will stop, acknowledge a whole bunch of people in my team, and thank you for letting me speak with you for the hour.
All right, thank you again, Nigam. So we are serving lunch out in the foyer, and there's networking reception until 3 o'clock. You guys are welcome to stay. But let's do one more round of applause for coming here.
Thank you.
Awesome. Thank you all.
All right.